Hi, welcome back to another Pharmacy Republic and Nebula Health video. My name is Shazli Hassan and I'm the leading functional medicine pharmacist for both Pharmacy Republic and Nebula Health. We're back with mitochondria. In a previous video, we learned how mitochondria produced energy in the form of ATP. In this series of videos, we're going to look at what happens when things go wrong with the mitochondria. When things go wrong with the energy creation within the mitochondria, then we open the door to a number of diseases. And today we're going to look at the big one. We're going to look at cancer. We're going to see how when the mitochondria don't work in the right way, we can open the doors to all forms of cancers developing. I'd like to start this video with a quote from the famous researcher Otto Warburg, who found in the 1920s that there could be a metabolic basis for the cause of cancer. He said, cancer, above all other diseases, has countless secondary causes. But for cancer, there is one prime cause. Summarized in a few words, the prime cause of cancer is replacement of the respiration of oxygen in normal body cells by a fermentation of sugar. So he discovered that the cancer cells used an inefficient method of producing energy. They used a fermentation process in the absence of oxygen to produce energy. They didn't use this aerobic respiration here, which is extremely efficient. They used a process outside in the cytosol to produce their energy, a process called glycolysis or fermentation. Unfortunately, a lot of the work that was done by Otto Wahlberg was ignored. With the advent of the discovery of DNA, a lot of attention was diverted to look for a genetic cause for cancer. However, a number of Wahlberg's students or students of students in America, there was the lab in John Hopkins University with Pete Pederson and Young Hee Ko, who did a lot of work on the metabolic cause of cancer, and with hexokinase 2 genetic mutation, which is present in all cancer cells. We'll come on to that in a little bit. And there was maybe about 2011, there was another researcher who wrote an excellent book, Thomas Seyfried, Cancer as a Metabolic Disease. It's a bit of a technical read, but then there was another author, Travis Christofferson, who wrote this excellent book, Tripping Over the Truth. This makes it a bit of an easier read for the layperson. Fantastic read. I read that in a day. It was inspiring. So going back to the Wahlberg theory about how cancer produces its energy. So cancer preferentially gets its energy in the absence of oxygen. It uses a process called glycolysis or fermentation, which occurs in the cytosol of the cells. So normal aerobic respiration occurs within the mitochondria, much more efficient, produces much more ATP. Glycolysis or fermentation, when it occurs in the cytosol, only produces 2 molecules of ATP, whereas if that molecule of glucose is burnt via aerobic respiration, inside the mitochondria, we produce a massive 38 molecules of ATP. So remember that in the cytosol, outside the mitochondria, glucose is burnt in the absence of oxygen. Inside the mitochondria, it is burnt in the presence of oxygen. So inside the mitochondria, more ATP, 38 molecules of ATP per molecule of glucose. Outside, in the absence of oxygen, we can produce 2 molecules of ATP. So what happens is when we eat food, we break that food down to glucose, and that glucose is present in the cell here. Then we break that glucose down into pyruvate. Okay, so that pyruvate then has a choice. It can either move into the mitochondria, go through the Krebs cycle, and then we get processed through the electron transport chain to produce ATP, as we've discussed in a previous video. Or it can stay in the cytosol, get converted to lactic acid, and in the process produce ATP. So it has a decision to make. Which way will it go? Will it go into the mitochondria or will it stay in the cytosol? In a normal cell, preferentially, we want to create more ATP for less work. So we're going to go inside the cell. The pyruvate is going to go inside the cell and get converted eventually to 38 molecules of ATP. In cancer cells, it prefers the inefficient process. And so one of the hallmarks of cancer cells is that there's a large presence of lactic acid, which actually leaks out of the cell and causes damage, and this facilitates spread of the cancer. So this begs the question, if this aerobic respiratory process is so efficient, why is there a need for the inefficient glycolysis? So this happens because when you need a rapid burst of energy, it takes time for this process to work. So you won't get that immediate fix of ATP. Whereas with glycolysis, you can. So imagine a scenario where you're in the forest, you're minding your own business, and then you spot a big grizzly bear who starts chasing you. So your first reaction is going to be to run. Otherwise, you're going to become his ATP, okay? So you run, you start breathing heavily because you want to get as much oxygen into every cell, especially the muscles in your legs, so you can run away to drive this aerobic respiration. So the glucose is getting converted via a 10-step process to pyruvate, which is working its way into the cell. So when we've maxed out our aerobic respiratory capacity, that's when that extra pyruvate gets diverted to a glycolic pathway. It gets diverted to undergo a process of glycolysis, so we can still produce extra ATP when we've maxed out here. So if you like, during these emergencies where we need that quick burst of energy, then that door to glycolysis is open to allow that excess pyruvate, which is not being used up in the mitochondria via aerobic respiration. It gets diverted to the cytosol, where we can still grab those extra molecules of ATP. And that process, that glycolytic process, is great for these short bursts of energy which require ATP. But as soon as we escape danger, we get to our car and we drive off and the grizzly's way back in the forest, then things return to normal. We close the door to the fermentation process and we go back to our normal aerobic respiration. However, in cancer, that door is left open. So the pyruvate preferentially goes down the fermentation pathway rather than move into the cell. One of the first steps in the pathway of glycolysis or fermentation is a transformation of glucose to glucose 6-phosphate. So usually in chemical reactions, there's a sequential series of minor changes to the molecule to get the end product. So if we start with glucose, we end with pyruvate. There's a series of 10 steps. The first step is the production of glucose 6-phosphate, and this is catalyzed by the enzyme hexokinase. In 1977, Pete Pederson in his John Hopkins lab discovered that in cancer cells, the hexokinase has been hijacked by hexokinase 2. In a normal cell, when the glucose is converted to glucose 6-phosphatase via the hexokinase, there's a negative feedback mechanism. So when there's enough glucose phosphate, it turns off the hexokinase. When we get this genetic mutation of hexokinase to hexokinase 2 in the cancer cells, the hexokinase 2 ignores the negative feedback system and carries on the conversion of glucose to glucose 6-phosphate. Despite there being enough, it will just carry on perpetuating this cycle. There's no off switch. The team at John Hopkins University, when they were working on the hexokinase 2, they realized that there would be an overstimulation of the hexokinase. It would not be subject to a negative feedback. They also found that it was bound to another membrane associated protein which acted as a channel. It was called the VDAC, voltage dependent anion channel. And what this did, this allowed molecules to flow in and out of the mitochondria. So essentially it played a crucial role in the regulation of mitochondria in a particular process called apoptosis, or organized cell death, or cellular suicide. So when a mitochondria has become dysfunctional and is inefficient and is creating a lot of free radicals causing destruction to neighboring cells, what happens is then that mitochondrion is programmed to die, and it does so by releasing this membrane-bound cytochrome C, which is released via that VDAC channel into the cytosol, where it's picked up, it's recognized by certain messengers who then initiate a cascade to organize the control death of this mitochondrion. If there wasn't an organized cell death, then all the free radicals and all the destructive elements within that mitochondria will be free to be released in an unorganized manner, causing destruction throughout the cell. So when the hexokinase 2 was bound to the VDAC channel, it closed it. So what this meant was, when the mitochondrion was malfunctioning and it wanted to initiate apoptosis or cellular suicide, the cytochrome C would dislodge from the membrane but would be unable to make its way out of the mitochondrion because the VDAC channel was closed. Okay, it would not be present in the cytosol. The cascade for cellular death or suicide could not be initiated. So by combining and joining forces and closing the VDAC channel, the hexokinase had turned this mitochondrion into an immortal cancer cell. Another interesting thing the hexokinase does is it attaches perpendicularly to the ATP synthase so that it can steal any ATP that is produced to further provide energy for this now immortal cancer cell. So to summarize, Otto Warburg first observed the phenomena of this metabolic dysfunction in cancer cells, where they preferentially used fermentation to produce ATP as opposed to aerobic respiration. Remember, they did that in the absence of oxygen. So they did that in the cytosol without the use of oxygen. So they didn't use the oxygen to burn the glucose. Okay, it's important to bear in mind that even though they did this in the absence of oxygen in the cytosol, that's not to say that oxygen wasn't present. It was. Yeah, it's just that the cancer cells just chose to use this inefficient process. Otto Warburg was one of the first to actually observe this process, and then he could actually see the buildup of lactic acid as a consequence of the cancer cell using fermentation as opposed to aerobic respiration. The researchers at John Hopkins University, Pete Pederson, they were the researchers who actually went further and expounded on Otto Wahlberg's observations. They discovered the how, as we've discussed earlier with hexokinase. The team at John Hopkins University discovered how the hexokinase was hijacked by hexokinase 2 in the cancer cells and then attached to the VDAC channel and also attached close to the ATP synthase and organized a kleptocracy where it grabbed all the ATP for use in this immortal cancer cell. So that's a brief story of how we came to know about the metabolic basis for cancer. One thing we know is that cancer has an incredible sweet tooth. It can only use glucose to produce ATP. Okay, so if we expound on this idea, how can we starve cancer? Okay, we can restrict the input of glucose. How can we do this? We can follow a caloric restrictive diet, Or we could do something that is quite popular at the minute. We could follow a ketogenic diet. How does a ketogenic diet starve a cancer? Well, for one, we're going to be using a different source of energy for our fuel. Remember, we need to get acetyl coenzyme into the citric acid cycle to produce these electron carriers, which then transport electrons to the electron chain. Okay, cancer doesn't care about this process. It simply needs glucose in the cytosol, where it can ferment this sugar to produce ATP. So if we remove glucose from the equation, or for example, if we remove carbohydrates from our diet, we force the body to use what's left, and the body starts to burn fat, and we use ketones as a fuel source, which cancer cannot use. So the theory is now that if we follow a ketogenic diet, we can actually starve the cancer. So one way I like to visualize all of this is that when the cancer cell uses the glucose as a fuel, it's as if it's using the old dirty diesel that we put into these old lorry that is spewing out loads of pollution, and we can use the pollution analogy as to the lactic acid that is as a byproduct from the glycolysis. When we compare this to the ketone bodies when used as a fuel, it's like electricity. So we're seeing these pollution-free electric cars. So when we force the body to use ketone bodies, we don't produce any pollution like lactic acid if we was to go down the glycolysis pathway as the cancer cells do. I hope this video has gone some way to help explain the metabolic basis behind the development of cancer and how we can help starve cancers by incorporating a ketogenic diet. We'll expand on this idea further in some more videos. We'll explain exactly how the ketogenic diet works in more detail, so you get some more like diagrams like this, I'm afraid, because we would like to get down to the science to understand what's really happening. So we'll explain how the keto diet works and what is a keto diet, and giving examples of what it usually contains, what are good fats, because in the past someone used to say, there's good fats and bad fats, and I was quite confused what's the difference between them. So we'll address this and much more. Again, so if you have any questions, please leave them in the comments. Give us a like if you like this video, and if you haven't done so already, subscribe to us. So that's it for now. I hope to see you next time, where we'll develop on the ideas that we've introduced here. Thanks. Bye.